Okay, welcome to OVA Hour number 9. I'm Jennifer Garrison. I'm a faculty member at the Buck Institute for Research on Aging. I'm also the co-founder and director of the Global Consortium for Reproductive Longevity and Equality. So at the Buck, we're pioneering a global movement that is aimed at advancing scientific research that's focused on female reproductive longevity. We think about reproductive decline as something that influences gender equality. Reproductive decline in midlife is about far more than just fertility or menopause. It's impacting a woman's entire life experience, career, family, health, well-being. So through funding and collaboration and hopefully innovation, we're trying to accelerate the pace of discovery towards diagnostics and useful products and therapies for women. So understanding how and why ovaries age prematurely, we think will also provide us clues for what's happening in the rest of the body. So as we're building this ecosystem around reproductive longevity and equality, I have the privilege every single day of talking with amazing scientists, clinicians, entrepreneurs, funders, lots of really exciting people. And OVA Hour is my opportunity to introduce you to them. So today I'm really excited to welcome Holly Ingraham. She has been working for over 30 years on female physiology and reproductive health. She's an international leader in hormone signaling, and she stands out really as one of the prominent senior women in her field. She had her PhD in physiology and pharmacology from UC San Diego, then completed her postdoctoral studies at the Salk Institute, and she has been faculty at UCSF since 1991. She's currently the Hertzstein Endowed Professor in Molecular Physiology. She's the Vice Chair of the Department of Cellular Molecular Pharmacology with a joint appointment in the Department of Reproductive Sciences. She is also director of the UCSF IRACDA Scholar Program to promote diversity, which we'll talk about a little bit later. She has won more awards than I can get to in the short time we have. So I'll just stop here and welcome Holly. Thank you so much for joining us.
Oh, thank you for having me.
Yeah, we're really excited to talk to you. I also failed to mention that you are one of our inaugural senior scholars for the GCRLE Scholar Awards. So you have a grant from us to study female reproductive aging, which is super exciting. So I wanted to start by just asking you a little bit about your work. So you've been studying sex-dependent mechanisms that the brain uses to control reproductive organs, essentially for your entire career. You started studying estrogen responsive circuits long before we thought about them with respect to aging. So can you tell us a little bit about your early work, and then maybe talk a little bit about some of the obstacles that have kept this particular field largely pretty much unchanged for the last 4 decades?
Yes. Okay. So, well, actually, when I first started at UCSF, I was really interested in how, the gonadal development was controlled, male versus female, and what were the molecular mechanisms that were responsible for that. And, there, in this area, there's a lot of work that describes wonderful morphology, wonderful descriptive studies, mainly focused on development, but very little mechanistic insight. And I would say that that is really the theme of this whole area, is that part of the reason that we have not understood as much as we need to understand, and we can't really tackle things at a really fine, a granular level in terms of therapeutics, is that we don't understand mechanism. And so we don't understand the basic molecular components, the molecules that are involved, the signaling pathways that are involved. And I think that has really kept the field where it's been for some time. And we can talk about sort of, funding aspects of that and why that might have happened. But my early work was really dedicated towards understanding the molecular mechanisms that contribute to the bifurcation of gonadal development. So why, testes begin to develop and females develop a little bit later. And I think throughout all of this, when I think about all of the work, um, in a way, we've been more focused on, maleness, because maleness actually, there are these adaptive responses in evolution that give us maleness, whereas, it's considered the default is femaleness. So
I think a
maybe,
lot of people don't realize that in biology, there
yes.
has to be a special, like a turn. There has to be a fork in the road to get to a male. But if nothing happens, the default is female, or at least, female as we define it in terms of
Right. So in a way,
chromosomes.
maybe the male bias in research is comes from this evolutionary perspective that maleness requires these additional steps and features, whereas femaleness doesn't. I was just recently talking to an evolutionary medicine person who really thinks that way.
Mhm.
So um, I think that uh, so yeah, there's been a lot of effort to understand maleness, and in a way, there has really been a bias in research by researchers to really simply work in males
Oh
versus
yeah.
females. So
It's been, yeah, it's we've touched on this a little bit in some of the other ovas, but most studies have focused solely on males, and that has really made, I think, the male body sort of biology's baseline in a way. And in those rare cases where females are included, nobody's looking at reproductive physiology,
no.
almost never. So all of that data we have around aging is in men. I think in recent years there's definitely been a change.
Mhm.
Maybe the last 10 years has focused more on women's health and focused on including women and females in female animals in studies. Um, Part of the reason that females were excluded was because the idea that their cycles, their hormonal cycles, were essentially contributing noise to the data, when in fact, that's the signature for female
Right.
health. Turns out to be sort of important.
That's
Um,
right. That's
yeah,
absolutely right. And I think that it's that complication scientists just basically didn't want to deal with. And we actually just today published a study on bioarchives. It was a study done with my husband and David Julius and Stu Brierley and Jim Bear, where we're looking at the cellular contributions to visceral pain. And
oh.
it's pretty interesting because my involvement in this study made them start looking at females.
You mean they
And
weren't going to otherwise, right?
well, maybe, but I said, there's no way I'm working with you guys unless we're working with females as well. And it
Yes.
turns out there's some pretty big differences. And of course, things like irritable bowel syndrome really affect many more women than men.
Mhm.
So, this is just, we have this, the way I look at it is, this is a great experiment between males and females, and nobody, you don't have to actually go out and knock out a gene or do anything fancy. It's just there in front of us, and we really just need to exploit this and leverage that great experiment in biology to understand more about female physiology and women's health.
Yeah, we need more basic research. We need more medical data for women, especially in that older phase of their lives.
Yes.
Women definitely age differently than men, but most of our data on aging is in men or in males.
Yes.
So can you tell us, so I haven't read the paper since it came out today, but sorry about that. I am familiar though with that line of research that you're doing. And I assume that this has something to do with the gut if you're
Yes.
talking about visceral pain. So can you tell us just briefly what you found and whether there were any sex differences?
Yeah, so that's really it's really interesting. And this was an amazing team, really. And so what we've identified are these specialized cells in the epithelium. They represent they're very, very sparse. Well, they're less than 1% of all the gut epithelium. And
So these are the cells that line your gut.
these are the cells
So
that
just
line
a
your
few
gut.
of those.
Just a few of those. And basically what they do is they talk to what we call mucosal afferents, so nerves that are going to go into the uh, dorsal root ganglion, spinal cord, and then send relay
So it's a
messages
direct route to the brain,
up to
essentially.
the brain. And so what we found is that basically you can manipulate the activity of these cells. So you can either silence them or you can really ramp them up. And what we found is that if you silence them, females really show this profound effect of silencing them. And then if you ramp them up, males really show an effect. So basically, what we're saying is that females already have this really heightened sensitivity versus males. And that heightened sensitivity really may explain some of the sex bias that we're seeing in irritable bowel syndrome, which is about a threefold sex bias. So it's really quite profound. And you can see it. Our collaborator did what we call intravital imaging, where you create a mouse where you can actually see the activity of these little signals up to the brain, and you can really see that in females they're just ready to engage, so that when you tweak them with an irritant like we use isovalerate, when you tweak them with one of these irritants, they're just ready to they're already ramped up and ready to go. And so it's really quite it's really fun to see this sex difference that
So
is.
we sort of already knew, at least I knew from my own experience, that I'm probably more sensitive to pain than many
Yeah.
of the men I know. So it sounds like you maybe discovered the mechanism by which that functions. So do you think there's a path then to maybe intervene in a therapeutic way in that
Well,
model?
I think that's really interesting. I mean, of course, we, as basic scientists, we always want to know more. So, of course, at this point, the people in my lab are really trying to understand, is this sex bias actually within these cells, or is it that circuit, that connection to nerves and up to the brain that's creating that difference in the sexes? So that's what we'll be working on. And
Okay.
to try to understand, but that's just one of the, that's just an example. And I didn't really want to necessarily, I mean, this work doesn't really relate to the reproductive biology, but I
Oh, but
think,
it's so cool. And it
I
does
think,
actually relate to women's health.
I think it illustrates the fact that, if you don't go looking for something, you won't find it. You won't find these really striking differences,
Yeah.
striking sex differences.
All right. So coming back to papers I have read from your lab, I just wanted to point out, so you talked about stimulating neurons, turning them on or turning them off. And those are tricks that neuroscientists can use now to really study circuits, so these groups of neurons that are either in different brain regions or in the brain and like you said, in the spinal cord and innervating peripheral organs that all are connected and talk to each other and control different aspects of physiology and behavior. And so, of course, we're both neuroscientists, so we're totally biased, but I think that the brain is a really key player for reproductive success, right? It really, those circuits really do ultimately control every aspect of female reproduction, from conception, fertility, menstruation, childbirth, lactation,
Yeah.
child rearing.
Yeah.
It's really all about the brain talking to the rest of the body, and particularly the reproductive organs, and that communication goes in both directions. So can you tell us, maybe start out by telling us the part of the brain that you're studying?
Sure. Yeah. So we study this area called the medial basal hypothalamus. So it's really sort of at the base of the brain. And it communicates, it has sort of this very cool positioning, because it's going to, parts of it are going to communicate to the rest of the endocrine system by talking to the pituitary itself is a really key feature for talking to ovaries and talking to the rest of a lot of other peripheral organs. So you have this nice anatomical arrangement between the hypothalamus and the pituitary that's
Mhm.
then sending out all of these really important hormones that are going to affect, for instance, ovarian physiology or bone physiology,
Mhm.
stress physiology, you name it. So that's the area that we work on. And we
And
work
it's
on,
part of the HPG axis. I think a lot of people will have heard of the HPG axis, and that first part of it, the hypothalamus, is what you're talking about.
we
I,
are, yeah. So the area that we work on probably doesn't directly communicate to the pituitary, but it definitely sends signals over to neurons that are going to then talk to the pituitary, the HPG, control the HPG axis.
well,
So
one of the things I think is so interesting about what you're doing is that I mean, we were just talking about the limitations and sort of the siloed nature of the research field over the last 4 decades. And I
mhm.
think part of that has been a hyper focus on the HPG axis and really not thinking outside of that. Well, it's clearly important. I think there's a striking diversity of neurons and circuits in the brain that may be overlooked and obviously are potentially important in terms of pathways of communication between the brain and the reproductive organs. And I feel like that's a place that you've really stepped up and made a lot of cool discoveries.
Well, I mean, if you think about it, I just in just, if you just step back and you go up to 30,000 feet, everything has been programmed for reproductive success. I mean, all of our physiology and all of our adaptive responses are really set up to try to ensure reproductive success. So um, it's I that's what I always get say at my seminars. And so, even when you're thinking about this total gut project that I'm just talking about, it's like, okay, so why do females, why are they more sensitive than males? I mean, why are they more sensitive why do they have this baseline sensitivity? It has to do something, it must all come back to reproduction.
Mhm.
It's what I always say. There must be some reason for this. So the
Can
brain,
I
yeah,
make a crazy suggestion? Just thinking off the top of my head, um, visceral pain in the gut might be a really important indicator of something being off with your microbiota, right? And we know that gut microbiome especially, can regulate circulating levels of estrogen, maybe the major regulator of circulating levels of
oh,
estrogen.
yeah.
So maybe it's more important that we notice that.
That's right. So yeah, so I mean, this is all to be worked out, but I think the brain, the hypothalamus, is really a key gatekeeper, and it coordinates all these adaptive responses to ensure reproductive success, and that's why we wanted to sort of really start studying that with respect to female physiology. And as you said, not be just completely focused on one set of neurons, but looking at this in a broader way. Because
Mhm.
I do think that even the just focusing on one set of neurons and having this dogma in the field that has been with us for some of the dogma of the HPG axis has been with us for 40 years, and nobody has really, people need to go in with new tools and new methods to say, okay, are these assumptions really right? And I do think there's some really great work that's coming out that's saying, hey, maybe it's a little bit more complicated than we thought, and maybe it's not quite, the dogma isn't quite as nailed down as it should be. So
Mhm. Yeah, I agree. So you have a lot of cool projects going on in your lab that I think are relevant, highly relevant to aging and fertility and menopause. You published a paper in Nature last October, which showed essentially that estrogen signaling to the part of the brain that you're studying can lead to increased physical activity in female mice, which is very interesting. So can you take us through that study?
oh yeah, that's so that was just a great study. It took quite a while, especially during COVID, because
Mhm.
we had to deal with mice and getting rid of mice and then having them come back online. But anyway, we got through it. I'm really happy to say we're re-emerging from COVID-19. Um, but so this study, what we had known that a certain group of neurons would control activity. We didn't really make the connection with how they could be controlled by estrogen.
Mhm.
But we knew that they were important, that these neurons were estrogen responsive. So what we went in to do is to really try to define what subset of neurons were controlling activity, controlling this aspect of energy expenditure
Mhm.
in females. And so we basically just through, some genomic types of work, omic studies, landed on a subset of neurons that express both, they respond to estrogen, but they also respond to the melanocortins, and they express the melanocortin 4 receptor. And this is another example where the bias for males had sort of excluded this whole region or these neurons because, in fact, in males, you don't see melanocortin 4 receptor being expressed there. Um, and people had ignored this area because it wasn't the main area that people had been working on. And that's because estrogen controls expression of this really potent signaling pathway, this melanocortin 4 signaling pathway.
So if I,
So,
oh, sorry, if I
yeah,
just to, for the people in the
clarify.
audience, no, just for the people in the audience who don't think about brain circuits every day. I've mentioned this before, but I think it's really cool and exciting. I study a different part of the hypothalamus, but all of the hypothalamus is super cool in that the neurons are all intermingled, and all of the things that we think about with respect to organismal homeostasis, it's all controlled right there. So energy expenditure is what you're talking about. And so the neurons that control reproductive function are right there, and sometimes the same neurons that control energy expenditure. So estrogen is made by your ovaries, and then it signals to receptors in the part of the brain that you're studying. And then tell us about what is melanocortin. And
Well, so melanocortin is very interesting. I should sort of back up and say that, the overall message of this story is that estrogen acting on this melanocortin signaling motivates females to get up and move. And
to get more active.
yeah, we become more active. And so it's all linked in with sort of this, what we call pre-ovulatory surge in activity. So if you, because I think it's really important to understand the basic physiology here. So if if you go through the cycle of the menses in our menstrual cycle of, and you look at activity, you can see this surge in activity right before ovulation. So again, why do you want to have activity before ovulation? Mating and reproductive success. So I go back to my main thesis that everything is set up for reproductive success, and you want to be more active right before ovulation to ensure reproductive success. So essentially what we're saying is that right before ovulation, you get this, we know that you have this surge in estrogen, and estrogen is then causing an increase in this whole melanocortin signaling in those neurons. And it's that process that is actually leading to the increase in activity. So what's important to note about our study is that we didn't put wheels in these little cages. These female mice just get up and they want to start moving. And so we could manipulate this whole signaling pathway by either ramping it up or suppressing it, and we got the appropriate responses where you get increased activity. So if, for instance, if you do 10,000 steps a day, you would suddenly be doing 20,000 steps a day.
Hmm.
If you
So there,
activate this full
okay,
time.
so these are the neurons then that are essentially, let's see, regulating the balance between sedentary behavior and physical activity. And these neurons are taking into account both energy state and
Yes.
reproductive cycle. Is
Right.
is
So
that fair to
it's
say?
really this blend where it's this convergence of basically, yeah, energy homeostasis and hormones.
Oh,
So
okay.
it's this beautiful convergence of these two signaling pathways to then say, okay, I want to get up and move. And of course, males can respond to this if we give them estrogen, but of
Mhm.
course they don't have lots of estrogen. And
Right.
so this circuit is really not active in males.
It's just a female specific circuit.
But we nevertheless, we can exploit, we can exploit this information and we can turn it on in males if we artificially tweak the circuit. So that's sort of cool. And I do think that, the, the, the scientist in my lab who was really the head of this study, Bill Krause, is now wanting to sort of manipulate this circuit to look in models of Alzheimer's disease
Mm.
to see if we offset, if we can improve brain health by turning on this circuit.
Wow, that is really exciting. This is obviously in mice, so there's a lot more to be
Yeah.
done. And then there's a lot of questions to ask in humans. Obviously, we're here to talk about reproductive aging. So in females, for sure, decline in activity is tied to reproductive aging. And after menopause, women do tend to be less active and they develop more fat mass and change their fat distribution. Um, I'm curious, so if estrogen is so tightly linked to physical activity in women, what about HRT? Does that
Yeah.
seem then like, I mean, I should say your article was profiled in the New York Times. And when you read through the comments from readers on the New York Times website, much of the commentary and discussion was around HRT.
Mhm.
And
Yeah.
it seems like women are just, desperate for information and trying to understand what the right thing to do is. And obviously neither one of us is a medical doctor, so we're not giving any medical advice here.
Right.
But the data that you collected and the work that you did suggests that maybe HRT could help.
Well, yeah, I think, if you look at, I mean, I think what you were saying about, as soon as estrogen declines for women, which begins at about what, 47, 48, and then, by 50, over
Oh,
50.
it starts even earlier. Yeah, but
Yeah,
by 50, it's gone.
and for some women, it starts much earlier. If they go into premature menopause, I mean, all of this occurs much earlier. And what I think it's really important to say is that all the breast cancer survivors, if they are compliant with their hormone treatment, which are these anti-estrogens, either by blocking aromatase or by SERMs that block estrogen, the estrogen receptor, all of those women, many of them are in their early 40s, are going to go into premature menopause
Yeah.
because essentially you're depleting all your estrogen. So it's not just women that go through natural menopause. It's all the patients and women that go through premature menopause. It's all those patients, millions and millions of breast cancer survivors that take these anti-estrogen or hormone treatment.
Mhm.
So I think what we're talking about is it's huge for women. This is a huge area of concern for women. So if you uh, and the other thing I want to say is that if you look at the data and you look at they measured a really important study measured visceral waist circumference and visceral fat after years after menopause, and they actually graphed it, and it's a 6% increase
So
in
bad.
visceral mass every year after menopause. So that's, now there's lots of women that are really fit and they work, you know, they either have really great genetic modifiers that we still don't understand,
Mhm.
or, you know, they work really hard at not having that happen. But these are just the data, the average data. This is pretty remarkable that you have this huge increase in visceral fat mass, increase in weight, and then along with that is this increase in sedentary behavior.
And I know that graph, it's horrifying. I can picture it, but
Yeah,
I don't remember, was that data entirely in women who were not on HRT, or was it a mixed
I
population?
don't know. It wasn't
I can't,
HRT. It's
yeah,
just,
that's what I thought.
yeah,
So it's without HRT. So
without
presumably,
HRT.
if you did the same measurement with HRT, it would hopefully be an improvement.
Yes.
We'll
Yes.
have to look for that data. It
Yeah,
might
look,
be out there.
I can send you the study because I was just curious about it, and it was it really was horrifying.
Yeah,
So,
we'll post it on the on the Juicy Early website
and of
for anybody
course,
who wants to be horrified.
yes. So, and you know, we are, you know, everybody's living longer, and we do want to age in a more healthy way, right?
Yes, that's what we're doing at the Buck
Yeah.
Institute. I mean, that is our entire goal.
Yes. So I think, you know, all of these aging institutions that are cropping up all, you know, they're springing up all over the place. I don't know what the goals are of these institutions, but I do think the goals should be, I mean, from a woman's perspective, I want to be able to age in a healthy way.
I
And
agree. Couldn't
so,
agree more. Yes, that's definitely our goal. I can't speak to any other institutions, but health span is what we call it. So just extending the number of years that someone's healthy. We don't really care so much about overall lifespan. We just like to keep
yeah,
you healthy until the minute you go to sleep and then
you know, I think
die.
that's just a fantastic goal that and you know, as a basic scientist, I can work towards that goal.
You are working towards that goal. Understanding how and why ovaries age prematurely is most definitely going to give us some clues about aging in the rest of the body.
Yeah. So
Yeah,
yeah, I mean, with HRT, it's, you know, again, I do think everything is sort of reflecting this male bias that we
Mm.
have in our health industry, not only our biomedical research industry, but our health industry. And, you know, the decision to sort of reverse course with HRT based on the nurses study uh, is, you know, it really should be challenged. And I know it has been challenged, but it has been challenged in academic medicine. But the question is it challenged out there with your private physician?
I'm not sure it's translated through. Actually, over hour number 2 was devoted entirely to going through the WHI study, breaking it down, and actually talking about what it shows, what it doesn't show,
Yeah,
and really trying to counteract some of that. I would call it misinformation that's
yeah.
out there about HRT. Certainly, it's true that there's data to suggest that you really need to start it within a certain window after menopause. That
Mhm.
that there's a sweet spot for when to start it. There's no data that suggests you should ever stop it. And but there is data to suggest that if you start it too late, that it could actually have a detrimental effect. So these are the kinds of conversations that you really should have with your physician. And one of the things we're trying to do right now is to put together a clinical advisory board who could, help to create some of these guidelines and maybe help us update the standard of care in a way that would,
That, you know,
put this information out there.
I think that if that comes together, that's such an important you know, that would be fantastic because, you know, we have all these boards for other things like you know, we have these boards debating whether trans can be in the Olympics. And I mean, they have all these national boards for issues like that. there really should be something for this that's affecting so much of our population. It would be wonderful to get clarity on this.
Yeah,
Um,
those are important issues too, and actually really interesting from a biological perspective. But I we may have gone a little off track here.
no, we don't want to go off track.
I don't mind. I'm sure the people watching don't mind either. It's really interesting what we're talking about. But there was another paper you published a few years ago, right before the pandemic, which was really focused on how the brain controls bone remodeling. And skeletal homeostasis is something that obviously we know is really impacted with aging. Osteoporosis, the risk of osteoporosis goes up dramatically after menopause. And so you identified this, again, a female specific brain, in this case to bone pathway that basically diverts calcium and energy stores away from bone building when energetic demands are high. So can you talk a little bit about that paper and that study?
That was such a fun paper, and I think we're finally have made some progress on figuring some stuff out. So we hope to be writing up the sequel to that story shortly. So this was a very unexpected finding. We were, again, as scientists, and I think this should be a life lesson, you find what you're looking for, but if you don't pay attention to data and what's around you, you will miss some things that are very, very important in life and in science. And so we were manipulating estrogen signaling again in part of the hypothalamus that we've already talked about, looking specifically for, well, it was really sort of a control experiment for this other region of the brain that we were looking at. And the postdoc at the time, Stephanie Correa, who now has her own lab at UCLA, just decided to follow her curiosity. And I would say that curiosity is a really important feature here of any discovery. So that's why it's important to have diversity, and we can talk about that in a minute, of individuals, because they're curious about different things. And women scientists like myself are curious about female physiology. So she decided to look at bone. And it's not something that you have to use a special machine to do that. And so she and it takes a little bit longer because it's just more involved. And she found this remarkable increase in bone mass only in female mice. And so we've now, we've figured out the neurons that might be controlling that. And again, if you go up to the 30,000 foot view of this, is why might that be happening? And essentially what we think is happening is that during times when you need energy to move around, to do other things for offspring, you basically don't want to put your energy into bone, because bone is energetically very costly and it's hard to get energy out of bone.
Yeah, it's an energy sink in a way.
It's an energy sink. So it's not like having energy stored in your liver through glycogen where you can quickly mobilize.
Grab it back.
Yeah. So we think that this whole system, that the brain is controlling bone remodeling, because it's basically saying, we don't want you to go and continue to form bone when we need energy elsewhere. And so what essentially we did is we sort of broke that whole circuit, that axis between the brain and the bone. And now the bone is saying, yeah, I'm taking all the energy and I'm just going to get stronger and more dense. And that's essentially what we did. We created these very dense bones. But of course, now we want to understand why these bones are dense. And so we have, stay tuned for this, but we know that when we manipulate those neurons in the hypothalamus, we are causing a change in a circulating factor that we may think comes from the pituitary. We're confirming that now. And we've done things like, I can't say anything about the factor yet, but
That's okay.
we've taken this factor, and our collaborators at Stanford, who are just fabulous, Chuck Chan and Tom Ambrosi, have taken this factor and put it on 70-year-old skeletal stem cells in vitro.
70-year-olds, 70.
70-year-old skeletal stem cells, and shown that you can get more mineralization. If you just put on regular media with nothing in it, nothing happens. But when you put on this factor, they start mineralizing and creating bone.
Cool. So this is a potentially a treatment for diseases that
it could be for
bone
aging.
loss.
So we're really excited. They've looked at fracture now with this factor, and again, it's increasing remodeling and bone repair. So we're really excited. And again, I don't think anybody would have really, the bone people never venture into the brain, and the brain people never think about bone. And so I
think about bone a lot.
Yeah.
But yeah, I think that we're in the minority, maybe.
Yeah, you do. But the problem is, when it comes right down to it, people like to stay in their lane, and they don't like to take that little road off, that country road off to another that will get you to another highway.
I think that's the best kind of science, discovery science, where you find something that you weren't looking for, and then it turns out to be something really important.
Yeah. So we're excited. We're just trying to get finished up with this study, and it's of course, you always go, is it right? Is it right? Do we have it right? But so we're working on it.
Well, I'm excited for that follow-up paper and to find out what this factor is.
Yeah.
And so then that brings me full circle. There's another paper you have on bioRxiv, which isn't necessarily related to female reproductive aging, but it talks about and describes some really interesting sex differences in liver function.
Oh, yeah.
And we know that liver is important for reproductive health, but you were doing, again, I would say a study on something completely separate, which was survival advantage that male mice have when you infect them with a lethal dose of bacteria. And then, correct me if I'm wrong, but you linked that to, you found that metabolic changes that are associated with type 2 diabetes and obesity are also linked to bone loss. Is that right? Did I get that right?
Oh, no. Well, it's not, yes. So this is a sort of Yanni Nicholson, who came from the Ajay Chawla lab, came over and started, he had looked at the susceptibility of males to infection, that they're very protected.
Mm-hmm.
And so I said, well, you really need to look at their metabolism and stress them out with a really high fat diet. And when we did that, all of a sudden you could see this huge sex difference in that males they really get fatty liver, they get diabetes, and females are completely protected from that. Whereas females completely drop dead with an infection. So there's this really big difference, this really big sex difference with respect to it. If you infect, we're talking about mice, you give them a pathogen. Yes. There's a huge sex difference between when the susceptibility of a pathogen for males versus females. But the flip side of that is that it's completely reversed when you stress them by making them eat high fat diet for 21 weeks. So, again, all of this has a lot to do with reproductive success. So females, again, we're talking about after menopause, you get all this visceral fat, but before menopause, all females, including human females, store fat subcutaneously and not around your waistline. And that's, of course, because again, it's reproductive success and progeny, is that you want to be able to mobilize that fat during periods of starvation or famine, or when you're, when your energy supplies are depleted. You want to get that fat back to you, back to your offspring, and back to you for reproduction. And
Right.
females just do that much, much better than males. And if you go and you look at all the data for in all of the historic famines, females have survived much better than males. So we sort of think that this is one of the reasons that you have this change, this distribution of fat change, but then female mice in this instance are much more susceptible to pathogens than males. And of course, I don't know, I mean, I feel like this is really hand waving, but males have to disperse more. They're going to have to fight for stature, fight for mating. They're going to be exposed more to pathogens than females. So in early days.
No, it's fine. These are hypotheses we can test, right?
Ukraine,
that's what we we come
if you
up
look
with.
at Ukraine, it's the men that are fighting. It's not the women that are out there fighting most of the time. So this may, unfortunately, this still exists. So.
Well, I don't want to end on a
No, we have to end
on a positive note. Well, I just wanted to say, I mean, again, I'm obviously biased, but I think that the work you're doing is spectacular, and I'm very excited to see what comes out next. I wanted to just tell everyone about some of the things. You're the lead organizer and director for two really important initiatives at UCSF that are focused on increasing diversity in science. So one is the UCSF Women in Discovery Science, and the other is this really unique and I think amazing program which could serve as a blueprint for how to do this in other places. It's called the Institutional Research and Academic Career Development Awards Scholars Program. Right. So can you tell us about those things? Like why are you doing this? And what gap are you filling, and what is the program, and how does it work, and how can people apply?
Well, so I think part of my motivation for doing some of this is that, I was in a field that was really dominated by men. And I used to go to meetings, and it would be just a group of men sitting around. And as I would tell somebody, I would go to those meetings, I would feel like I'd have to put on my armor to go in and start having
bring your shoulders up.
Shoulders up.
I know that feeling.
Yeah, you know that feeling. And have these conversations with this group of men and hope that you would get the respect and that your science would get the respect. And of course, as a female scientist, because I really am interested in female physiology, right away, 20 years ago, that was like, well, why are you interested in these sex differences or sex differentiation? It's like, you're not interested you're not sort of being macho enough with the guys. And so, that I sort of endured that system for 20 years or so. And about 10 years ago, I just said, wait a minute, this just isn't right. And there were just very, very few women around me, and very few women role models that I could look at in my field. There just were none. Mm-hmm. So I said, this has got to change. How can we have this workforce, 50% of the graduate students, 50% of the postdocs were all women, and yet we're just completely losing them. And it's in part because they would go to meetings like the ones I attended, and they would feel like they were in a fraternity house or something. Mm-hmm. They just didn't feel welcome, and they didn't feel respected, and they only felt like they were given talks because maybe they were the token women.
Women, yes.
And so I just felt like and I would just have these horrible feelings sometimes, like just horrible, horrible feelings, even though I love my science, I love what I do, but I would have these feelings that I just didn't really belong. And I just decided that cannot happen anymore. So I
Yeah.
I gave a talk and I listed all of the things I had endured. And I said, all of you old people can get out of the room. I'm just going to talk to you young people. I want you guys to change the system. I want you guys to think about the fields that you go in. I want you to hold these people accountable because you have the power. You're doing all the work. And so I decided at UCSF, to come back and look at the data for women and was
Mhm.
sort of amazed that, it
Mhm.
wasn't looking very good.
I remember when I was a graduate student and I was there from 2001 to 2007, there were no female chairs or vice chairs of any department that I interacted with, not a single one. There was
Yeah.
no female in any position of leadership.
Right.
None. And it didn't even occur to me at that point that it was unusual, right? It like at that moment, it didn't even register with me.
It's so true how we're just such, I guess we're just such good little soldiers, we just we're doing all this work and we're going, wait a minute, why is the top looking like it is? Why are the area, all the leaders are men. So anyway, that clearly, with a lot of other things going on in our political environment, I do think, things are changing. And I think there's this real awareness now. But, it has to be, people have to continually bring this up.
Oh yeah, I know. I mean, for the consortium, one of the foci that we started with was to find novel ideas for how to keep women in science, right? You mentioned that there's 50% women at the grad school level, there's 50% women at the entering postdoc level, but then past that, the numbers haven't changed for decades. And so there's still something broken. We fixed the early part, like the leaky pipeline all the way up through graduate school is pretty much fixed, at least in the US. But what's happening after that, the numbers for women in leadership roles in academia, are like 27% or something like that.
Right.
And that's been true since the 60s.
Yeah.
So coming up with novel ideas for how to fix that. We can talk more about that later.
Right.
But I want to hear about this. Can you tell us just a little bit about the program, just a snippet, and then we'll link to the website.
Okay, so
on our
this
website.
is, this is a national program. It was really started by NIGMS. And the idea was to basically provide the funding for trainees, scholars that come from diverse backgrounds, just all sorts of diversity. Okay, we're not just talking about skin color, we're talking about diversity, real diversity, diversity of experience. And to bring them in, give them the money, and create the community that makes people feel like they belong and makes people, empowers them and gives them strategies for increasing their confidence. So helping them with grants, helping them prepare for giving oral presentations, basically doing what kids from academic parents have, right? I don't come from a family of academic parents.
Nor do I.
And my husband doesn't, who won the NOAA. And so you have to have these support systems. And really, this is just a formal way to really create that.
Yeah, it's so important. Thank you for doing that.
Okay.
And I honestly think it really is a blueprint for how to do this in other places. And it's so timely and it's so necessary. It's so needed. We are up against the clock here.
Right.
There's two questions, which maybe we could start with the first one and see if we get to the second one. One person asks, do you have any advice for women looking to be successful like you in a male-dominated workplace or field? That might take more than a minute to answer.
Yeah, it'll take more than a minute. But I would say just, you have to be yourself. And women should not have to change their brains to exist in this structure.
I would also say so important to find female mentors who have been successful, and look to them for advice. The other question was about the science. The question is, with the mice that you were able to increase the bone density for,
Yeah.
were there any negative effects of that? It seems like that would be an amazing thing to do. Is there any downside?
Well, I think that's a great question. And, it's what you go looking for. And I'm sure, yes, I mean, when you create this, of course, you lose, you screw up the ovaries,
Mm,
because you're sort of screwing up that HPG axis.
mm,
So, of course, systemically, if we were thinking about this for a therapy, it wouldn't be something that you could just, you know, it give all the time, because I think you would screw up reproductive capacity. Now, for a postmenopausal woman,
mhm.
that would not matter.
Right, right.
Uh, or for men who suffer from osteoporosis, that would not matter.
Mhm.
So, again, we it's like we haven't looked for everything, and so I'm not going to say there aren't some negative side effects,
Yeah,
but
that's
uh,
fair.
yeah,
Fair. Yeah.
yeah.
Thank you so much for joining us. This has been a really, really interesting conversation, and I appreciate your time and
Okay.
everything that you're doing. We'll be looking for more exciting work from you and your lab.
It's all because of the people in my lab. All these projects are because
Of
of them.
course. Yeah. Yeah.
Okay.
Anyway, thank you all for joining us on OVA Hour, and we will see you again next month.
Thank you for being a great host.
Oh, yeah.
Okay.
Anytime. Okay.
Bye.
Bye, Holly.